Good evening, everybody. Am I audible? Good evening, everybody. Am I audible? Loud and clear? Loud and clear? Okay. So, are you all ready for the rapid revision rounds? Yes. Nikhil? Okay, good. Kantimati audible, fine. Okay, Yashira. So, I see a lot of yes coming up. Perfectly audible. Good to see you, Mahima. Okay. So, can I see a BIO in the chat box? Hello, P. Dubey. Hello, everyone. Yes. Okay. I see a lot of BIO coming up. Right. So, as you all know, today we are here to discuss some of the very important and very tough questions. Okay. So, that is what is rapid revision rounds. Okay. And I'll be discussing questions from Unit 2. So, as you all know, Unit 2 is cellular organization. Right. So, in cellular organization, what all comes? What all are the various topics, like from where the what is cellular organization? So, as you all know, okay, so it has, I'm sure you all know about the syllabus, and then it has various subunits. So, it has 5 different subunits. So, basically, the questions that we are going to discuss will be from the subunits, like about the membrane structure, function, the organization, the structural organization, and function of intracellular organelles. Okay, then organization of genes, and then very important, cell division and cell cycle. And then, yes, we have microbial physiology. So, before I start with the rapid revision, so I want to ask you all, what do you all think about the questions that come from Unit 2? Is it easy? Is it difficult? Is it very difficult or very easy? What do you all think? Moderate. Okay, Nida. Good to see that. Moderate, good. Yes. Average. Micro part is easy for you. Good. Okay. Difficult. For some, it is difficult. Kritika says difficult. For some, it's medium. Good. Shalini. Yeah. For many of y'all, like Ankita, Janvi, many of y'all are saying it's moderate. Yes. We'll come to it. Like, we'll discuss the questions, and again, I'll ask you at the end. So you'll get a better idea whether it is easy, difficult, moderate, right? Okay, Kaushik says it is like it traps you. Okay, but yes, how can we come out of it? By definitely by doing some, question solving, and that is why we are over here. So let's get started. So the first question is from the membrane structure and function, the very first subunit only. So it says lipid rafts are involved in signal transduction in cells. Rafts have composition different from the rest of the membranes. Rafts were isolated and found to have cholesterol to sphingolipid ratio of 2:1. The estimated size of the raft is 35 nanometers square. If the surface areas of cholesterol is 40 angstrom square and sphingolipid is 60 angstrom square, how many cholesterol and sphingolipid are present in one raft? Okay, so there are 4 options. So you can see the options, and now you have to solve this particular question. This is the first question, and let me see who can answer this correctly. Okay, Patel, cell cycle is difficult. We'll also solve some questions from cell cycle. Yes, Antara, agree with you. Yes. So now let's see. This particular question. I'll give you 2 minutes time to solve it. Okay, some of you all are answering. Good to see that. Yes, Kritika, Sunanda, Ramesh, they are answering the question. Okay, so what about others? Yes. Okay, some said option 1 is the answer. Many of you all said 3 is the answer. Ashwarya says 2. So I see a lot of options you all have selected. Okay, Mahima says 3. Is it? Are you all just guessing or are you all solving and telling me? Solving? Okay. Nandini, Kantimati, okay, good. Okay, okay. So I'll just wait for 1 more minute. Other students are answering. Solving. Good, Nandini. Good. Yes, I can see that you're solving. No idea. Okay. All have same ratios. Okay. So you have to find, we'll find it out. Okay. The ones who are feeling that you don't know. So we'll see how to solve this particular question. Okay. It is a December 2015 question. So you can see what is the standard of the questions that they ask. Okay, Nikhil, good. You're solving. Good to see that. Okay. So I'll just wait for a few more of y'all who are answering. Some are still solving. Okay, Nikhil, waiting for you. Yes, so now let's see what is the right answer for this particular question. So the right answer for this question, it will be your option 1. Okay, so first let me, like many of you all might have the doubt, like what is a lipid raft, right? So I'm sure many of you all might be thinking that. So if you see, this is what is a lipid raft. Where you find this lipid raft? Lipid rafts are actually some micro domains. So where you'll find it in the membrane, in the plasma membrane, right? So in the plasma membrane, you have these micro domains. They are rich in cholesterol and sphingolipids. So that is what is the question asking, right? So it has already mentioned about that. And then what it says, that they have cholesterol to sphingolipid ratio of 2 is to 1. Okay, and the size of the raft is given, and the surface area of cholesterol and sphingolipid is given. Okay, Ankita, not able to solve. Let's see how to solve. Okay, so have a pen and paper, and if you don't know, you can write the solution. Okay, how to solve this particular question. So first, how to go about this question, because they have given a ratio over here. So first thing what we learned in our mathematics class, let the number of cholesterol molecules, okay, because it is 2 is to 1, so we'll consider it as 2x, right? And because it is 2 is to 1, so sphingolipid molecules is, we'll consider it as x. So cholesterol will become 2x, right? Now total area that is occupied by cholesterol is, they have already mentioned, right? It is 40. Okay, now when you multiply, because we are considering it as 2x, so we'll multiply this with 2x, so it becomes, this is surface area whole square, so it becomes 80 angstrom whole square. Similarly for sphingolipid, it is 60, so 60 into the this x value, right? X, and then this is whole square, right? So this becomes 60 angstrom square. So we have just one x over here. Here it is 2. So you multiply, it becomes 80x over here. Here it becomes 60x. Now the total area of the raft is given. It is 35 nanometer square, right? So you have to convert it, because you can see the units over here is different. It is angstrom, okay, and here it is nanometer. So what we do, we convert it into angstrom. So you get the value of 3500, right? And then the total surface area is 3500, and then when you add this, that is 80 x plus 60x. Now you need to solve this particular equation. Clear? So when you solve this equation, you'll get the value of x as 25. So if x is 25 means the number of sphingolipid molecules is 25. So the cholesterol will be 2 into x. So it becomes 50. Yes, Mahima is right. So who all answered Nikhil correct? Yes, you were solving. So I could see you have answered it correct. Right? Everybody got it? Should I move ahead to the next question? Clear for everyone? Okay, let's move on. Let's see our next question. So it says the cell membrane of neuron maintains intracellular conditions that differ from those of the extracellular environment. Such difference in intra and extracellular conditions are critical to the function of the nerve cell. As the nerve cell membrane resembles a charged capacitor, assuming the electric field E across a parallel plate capacitor is uniform, and if membrane thickness is 7 nanometer and potential difference across the membrane is minus 60 millivolt, calculate E of the membrane. So find out which is the correct option over here. So this question is again from membrane structure and function. So in that membrane structure and function, we have a topic electrical properties of membrane. Okay. So many students tend to skip that, but you can see they have asked a part C question. Okay. And it is an easy question, just that you need to know what is the formula by which you can solve this question. And I'm sure you have studied this and you know the formula. It's an easy formula, and I'm waiting for your answers. So kindly solve and tell me what should be the right answer over here. So you have to think what is the formula, right? For, if you remember, E is equal to V by D. Okay, that is the formula. So you can use that formula and try to solve this particular question. Whoever does not know, you can use the formula E is equal to V by D. Okay, the formula is easy, but again, what you see over here is the units are different. So first you need to make all the units, no, you have to use the same units, and then you need to solve this particular question. Okay, some said 2 as the answer, some said 3 as the answer. So we'll see. Okay, we'll try to solve it and try to find out what should be the right answer over here. Okay. Okay, Lakshmi says 3. Okay, many of you all have said the answer as 3. So let's see what is the right answer. Okay, solve, Lakshmi, good. Okay, so still students are solving. Good to see that. Okay, Nikhil, Mahima, good. Yes. Yes, so now let's see how to solve this particular question. So students who are doubtful of this question, you can go through this whole of the question. You can write it somewhere and practice questions by using this formula. Okay, so here what is given is, if you consider the 2 points A and B, the potential difference is VAB, right? Because we're considering these 2 points A and B. And D is the distance from A to B, and E we'll consider it as the electric field, right? So now, as I said, the formula, you can use it, E is equal to V by D, or V, the voltage or the potential difference between the plates, is equal to E into D. Okay, it's the same formula. So here when we have to calculate E, so V by D, so V becomes 60 millivolt by 7 nanometer. So you convert this into volts. Okay, so 60 into 0.001, okay, becomes, so millivolts into volts, you have to divide by 1000, right? And then again, nanometer, you can convert it into meter. And then the value that you get is 8.6 into 10 power 6. Yes, yes, Ankita. Yes, right. Rimjim, right. So the right answer over here is your option 3, 8.6 into 10 power 6. Right? Now let's move ahead and see our third question. The frequency of cells in a population that are undergoing mitosis, we call this as mitotic index, is a convenient way to estimate the length of the cell cycle. In order to measure the cell cycle in the liver of the adult mouse by measuring the mitotic index, liver slices are prepared and stained to easily identify cells undergoing mitosis. It was observed that only 3 out of 25,000 cells are found to be undergoing mitosis. Assuming that M phase lasts 30 minutes, calculate the approximate length of the cell cycle in the liver of an adult mouse. Okay, so there are 4 options: 76 hours, 50 hours, 42 hours, 21 hours. So solve this question and tell me what should be the right answer over here. Easy? Is this question easy or difficult? Solving, students? Okay, solving, trying. Good, good, good. Shawnee. Nikhil solving, good. Mahima, so all are trying. Good, good to see that. So I know it's not a very easy question, as I see you all are taking time to solve, but yes, you are, if you know what is mitotic index, how to solve this particular question, then it would not be very difficult, right? Then it will be a wise decision to opt for this question and try to solve it and get the right answer. Okay, some said 2, some said 4, so I get many options over here. So still students are saying option 3, 4, 21 hours. Okay, Lakshmi. Okay, let's see what others are telling. What should be the right answer? "42 hours." Okay, 4, 21 hours. Okay, Lakshmi. So try solving and tell what should be the right answer. Okay, some said 3. Lakshmi, you're telling me the formula for mitotic index, but the question is asking mitotic index, you know how to solve it. Okay, but you have to find out the length of the cell cycle, the whole of the cell cycle. Okay, mitotic index, that time is only for, it's the time for the mitosis, not for the whole cell cycle, right? So you need to solve after that. After you have used the formula, so after that there is few more steps you need to do. Okay, Lakshmi, got it? Okay, Nikhil, Sangeeta have answered. Okay, we'll see what is the right answer over here. So mitotic index, as I said, what it is the sum of all cells in phases, as so like in mitosis. Okay, so we have different phases: prophase, metaphase, anaphase, telophase, right? And then you have to divide it by the total cell number, and you multiply it with 100, so you get the mitotic index in percentage. Okay, Janvi, now you're thinking the questions are not moderate. Yes, it's not moderate. But yes, if you know how to solve, it is not very difficult also, right? Okay, let's see now. So, mitotic index, I told you what is the formula. It is the total number of cells in mitosis by total cells. So, they have given what is, it was observed that only 3 out of 25,000 cells are found to be undergoing mitosis. Means the cells that are undergoing mitosis is 3 in number. You divide it by 25,000 and you multiply it with 100. So you get the mitotic index as 1.2%. Okay, Sunanda. Okay, fine. But once you see the, how to solve this question, it will not take time, right? Now, once you get the mitotic index, so from the cell cycle, what we find, if this is the mitotic index, means 1.2% from the cell cycle is in mitosis, right? And the rest will be obviously in the interphase. So the whole of the cell cycle includes interphase plus the mitosis, right? And interphase means you have the G1S, right? You have G2. So this is coming under your interphase. So for mitosis, it is 1.2%, right? Now 1.2% of the cells, okay, that is the mitotic index, is taking 30 minutes, right? Now so for 100%, because the total now, from 100% of the cells, 1.2% is in mitosis, then definitely the remaining is in interphase. So when we consider them as a whole, right, as 100%, okay, so what you have to do over here, you have to just use this unitary method and you have to find out 1.2% is 30 minutes. So 100% will take around 2500 minutes, and then you have to convert it into hours. Clear? So 1.2, you can write it like this. Okay, 1.2 takes 30 minutes. 1.2 is taking 30 minutes. So for 1%, it will take 30 by 1.2, right? And for the whole of the 100, you have to multiply. So I'll just write over here 30 by 1.2 into 100. So when you solve this, you will get the value as 2500. Clear? Did you all solve this? Did you all get this? And then convert it into hours. Okay, divided by 60, you will get the answer as 42 hours. So the answer is your option 3. So it's a December 2015 question. Okay, Lakshmi got it. Okay, Shionli got it. Sunanda, are you able to get it? Okay, Nikhil, clear. So let's move ahead and see the next question. So then the next question says, using FRAP techniques, diffusion coefficient of 3 integral membrane proteins M1, M2, and M3 in a kidney cell is calculated as 1 micrometer per second, 0.05 micrometer per second, and 0.005 micrometer per second respectively. Considering fluid mosaic nature of biological membrane and relationship of structural organization of integral membrane and protein with diffusion coefficient, which proteins will have highest number of integral domains? So you have four options. First M2 and M3, second M2 only, third M3 only, fourth M1 and M3. So think properly. Here you need not solve anything, right? You have to just apply whatever is given and you have to tell me what is the right answer over here. Okay, Sunanda, good to see that you got it right. Okay, Finansri, Vishal, okay. Okay, some very quick answers I am getting. 2, 3 students have already answered. What about others? I'm waiting for your answers. Okay. Okay, Kantimati, Hiral, Sandhu, good. Nandini, many of y'all have answered. Yes. So let's see what is the right answer for this particular question. Okay. So the right answer for this question is your option 3. So who all answered option 3? So you all are correct. Yes. Now, before that, for many of you all who don't know what is this FRAP technique, so I've just shown a figure over here that will help you to understand better what is FRAP. So it is basically fluorescence recovery after photo bleaching, right? So what happens, how they came to know that the lipids show lateral movement, okay? So the lateral diffusion rates of lipids were measured by using this FRAP technique. So here, if you take a cell and then you add certain fluorescent probes on the lipids, so what will happen? You react the cell with this fluorescent probe, so it will label these lipids. Okay, and when you observe it under a microscope, so you'll see the fluorescence orange color. Okay. Now, what you do is for certain time you use intense laser beam, so it will bleach wherever the beam strikes the membrane, so it will be bleach that particular membrane region, right? So what you will see now under the microscope, you'll observe a white patch because that region, the fluorescence has been bleached, right? But after certain time, what happens again, if you observe, so you will see that that white patch has been covered up, right? So what we conclude from this particular experiment of this particular technique is these lipids, okay, they show lateral movement and they can move from their position to the adjacent sides, right? Because they're showing lateral movement. So that is how the measurement of lateral diffusion rates of lipids can be observed by using this FRAP. Clear? Okay, now the question is about FRAP, and there they have mentioned that now here instead of lipids, it is they're also talking about the proteins. Okay, so proteins can also show this diffusion. So there is the diffusion coefficient values that is given. Now diffusion coefficient values, when it is given, so if the diffusion coefficient is more, okay, so when diffusion coefficient is more, so the rate of diffusion will be high. So high will be the diffusion coefficient, high will be the rate of diffusion of molecules, right? But now what is happening in the question, they are saying that you have to find out the protein which has highest number of integral domains. Okay, so the ones that is showing least diffusion coefficient, right? Least diffusion coefficient is M3. Least diffusion coefficient means it is showing least diffusion, right? Least diffusion. So in that case, what is happening? In that case, you can say that the proteins that will have high number of integral domains. So if you consider this is a membrane and there is a protein and it is spanning the membrane. Okay. So if it is spanning the membrane means it is not that free, right? It cannot move freely, right? So what is the right answer over here? Which protein will have highest number of integral domain? It will be option 3 because that has the least diffusion coefficient. Clear for everyone? Anybody has any doubts in this particular question? So proteins having high number of integral domain will diffuse slowly as domains will restrict the rate of diffusion, while the proteins having less number of integral domain will diffuse faster. Restricted lateral movement means they cannot move, right? Because they are, see, if it is a integral membrane, the integral domains means the protein is spanning the membrane. Okay, if it is a peripheral protein, it does not have an integral domain, so it will be just over here or over here. So in that case, it can easily move, right, from one lateral movement. Okay, the sidewise movement, either in this left side or right side. So that is the lateral movement. So, as your question, you're asking restricted lateral movement means it cannot move, right? Because the domains are integrated, they are inside the membrane, right? So, it will find difficult to move. But if these domains are not there, if the protein is free, just like here it is on the inside or on the top, like it is a peripheral protein, so it can easily move on the membrane. Got it? Lakshmi? Why not option 1? Because you have to find which protein will have highest number of integral domain. Where will be the integral domain will be highest? Nikhil, where the diffusion coefficient is least. Least means less, सबसे less, right? So those proteins which have least diffusion coefficient means they are not able to diffuse properly. They are not able to show lateral movement. So when they are not able to show lateral movement means what we can say that they have more of integral domains. That is why they are not able to freely move. Okay, M2 and N3, yes, M2 can also be an answer, but the question says which protein will have highest number. So M2, their diffusion coefficient is not same. If it was same, then we could have said that M2 and M3. Right, Nikhil, do you agree? Because the diffusion coefficients are different here. It is not the least diffusion coefficient. Clear? It is 0.05. Here it is 0.005. These values are not same. Got it? Okay, Nikhil. Answer lies in the question only. Yes, Debanjan, good. Highest number of integral domain. Yes. Exactly, Mahima, you have got it right. Okay, highest. Yes. Yes, Nikhil. So now I'm sure you have got how to solve this particular question. Okay. No doubts. Should I move ahead? Can I see a BIO? Okay, Ritika, fine. Let's move ahead. Okay. So you are studying the binding of proteins to the cytosolic phase of cultured liver cells and have found a method that gives a good yield of inside out vesicles from the plasma membrane. Unfortunately, your preparations are contaminated with variable amounts of right side out vesicles. Nothing you have tried avoids this contamination. Somebody suggests that you pass the vesicles over an affinity column made of lectin coupled to Sepharose beads. What is the rationale of this suggestion? Right side out vesicles will be lysed by lectin coupled to Sepharose beads. Right side out vesicles will simply bind to lectin coupled Sepharose beads. Third, lectin will bind to the carbohydrate residues present only on the inside out vesicles. Fourth, lectin will bind only glycoproteins and glycolipids present on the inside out vesicles. So I'll give you some time. I'll give 1 minute. You can just tell me what should be the right answer over here. Okay, some students are answering. Okay, I got varied answers. Some said 2, some said 3. Think properly and tell me what should be the right answer. Okay, 2, 3. So you're getting confused between 2 and 3. 4. Okay. Kritika says 4. Okay, we'll find out what should be the right answer over here. Okay. Okay, so you're studying. Okay, I'll just tell you first. The question is saying you have found a method that gives a good yield of inside out vesicles from the plasma membrane. Okay, so inside out vesicles means I'll show you a figure over here. Okay, so if this is no, they have given an example of RBC. So when we talk about inside out vesicles means the inside will be outside. So this is how inside out vesicles look. Okay, and if it is a right side out, okay, but in the question it is inside out, so inside out means inside should be outside, but if it is a right side out vesicles means it will be the same way, like the one that is outside will be outside, right? So this is what it means by inside out vesicles, means the inside will be outside. So whatever is inside, okay, whatever proteins, whatever lipids, so it will come on the outside, right? Now you are doing this, you are studying the binding of proteins to the cytosolic phase, and you have found a method that gives a good yield of inside out vesicles, but you find that there is a contamination with some amount of right side out vesicles. Okay, so now you know what is inside out and what is right side out. So you have tried avoid this contamination. Somebody suggests that you pass the vesicles over an affinity column made of lectin coupled to saccharose. Now you should know what a lectin does. Okay, first, lectin will bind to carbohydrates. Okay, so when lectin binds to carbohydrate, if you know that much, then it is a very easy question. Lectin binds to carbohydrates. And where do you find the carbohydrates in a cell membrane? Where? Where in high amounts? In the outer surface of the cell or inside in the cytosolic phase? Where do we find? In the outside, right? Okay. So that is where you will see the glycocalyx, that is your glycolipids, okay, glycoproteins. So when we talk about glyco means we're talking about the carbohydrates, so they are present on the outside. So now what will happen, the lectin will bind to the carbohydrates and carbohydrates are present on the outside. So in this way, right side out vesicles will simply bind to the lectin coupled saccharose beads. So in this way, what will happen, the right side out vesicles, that is your contamination, will be removed. Clear? Did you get this? So the right answer will be your option 2. Any doubts over here? So it is like a question that where they have coupled your unit 2 with unit 13, which is related to affinity chromatography. So it is basically affinity chromatography where they're using lectins. Okay, so lectins are carbohydrate binding proteins. Outside. Okay, yes, Nikhil, Nandini, right. So carbohydrates are more on the outside. So that is why the answer is your option 2. Now let's move ahead. Yes, so I want to also discuss about the students who are feeling you're feeling the questions are not moderate, you're not able to solve. Okay, don't be disheartened. Okay, Biotecnika has come up with the Lakshya batch, which is starting from today onwards. So it is no, you still have time, you can join this batch and you can prepare well. So now you know what is the standard of questions that are being asked, so it is very important that you make a very directed and in a right way. You need to do your preparation every day. So I'm sure who all are feeling that it is not very moderate questions, you can anytime contact with Biotecnika and you can join this batch where we will try to cover all the various topics from the various subunits, and you will get to know more of the difficult questions and the different topics in details. Okay, now let's move on to the next question. Following is the domain organization of 3 proteins that are targeted to the mitochondria. Based on the domain organization in the above figure and assuming the left box to be having the mitochondrial sorting signal, predict the most likely subcompartment of the mitochondria in which the protein will be found. So you have these 3, the figures, the domain organization. So based on this, you have to tell where will A enter, where will B, where will C enter. So try and tell me, quick. You have 1 minute time. There's nothing to solve. You have to just see the figure and you can easily tell me what is the right answer. It's a December 2015 question. Not very difficult, I'll say, but yes, you need to know the figures in details. If you have studied the carb, so you should know the figures. Okay. Okay, Nandini, you got confused. So I'm sure now you are able to solve the previous question properly. Yes, Ankita, right. Yes, Sean Lee, you are right. So they have given you the ones who are not able to read, I'll read it for you. For A, it is written matrix protease cleavage site. For B, it is matrix protease cleavage site, inner membrane protease site, and hydrophobic region. So this is about B. And in C, it's just written interacts with outer membrane import pore. Okay, and for A, it is matrix protease cleavage site. Okay, again I get varied answers. Many of you all said one, some are saying two. Okay. Okay. So let's see how to solve this particular question. Now, for this question, as I said, you need to know the figures properly. Okay, so I have just shown here one example, okay, for how the various proteins, okay, this is for mitochondrial proteins. So you'll see that the proteins that are entering into mitochondria. Okay, so mitochondria has a outer membrane, it has an inner membrane, it has an intermembrane space, and it has the matrix, mitochondrial matrix. So it depends what type of sequence, like that sequence will act as a signal, and it depends based on that sequence whether it enters into matrix or inner membrane. Okay, so if the first one is showing, now if you go by the question, so the first one is matrix protease cleavage site. So it's already mentioned it is a matrix protease cleavage site. So definitely the protein will enter into matrix. Okay, so you can see that in many options they have given matrix. So this is one thing that is clear, that the A should go into matrix, right? So if you follow this figure also, so you will see it has a matrix targeting sequence, so it will definitely go into the matrix, right? Then next is B. So again it has a matrix protease cleavage site, inner membrane protease site, and it has a hydrophobic region. So as it has a hydrophobic region, so when this hydrophobic region is there, so remember, whenever this hydrophobic sequences are present, so it acts as a stop transfer sequence. Okay, so when stop transfer sequence is present, so the protein will not enter inside the matrix. Okay, it will stop in the inner membrane. Clear? So once the cleavage site is cleaved, as you can see over here, okay, so I'm not going to not discuss this figure in details, just that according to the question you can see that it also has a matrix targeting sequence. So that sequence has been cleaved, and then it also has a hydrophobic or the stop transfer sequence. So it is a hydrophobic sequence, so that is why the protein cannot enter more inside the matrix. Now the protein will be there where in the inner membrane. Clear? Okay. Yes, Mahima, right. So hydrophobic region is present means it will go into inner membrane. Okay, and the C, it interacts with outer membrane import pore, means it will be on the outer membrane. So it will interact with the import pore on the outer membrane. So it means that this particular protein will enter into the intermembrane space. Clear? Got it? So the right answer is your option 1. Now let's move ahead. The next question says a single protofilament of microtubule grows at the speed of 2 micron per minute. Considering that there is no catastrophe in the microtubule nucleation and the size of the tubulin unit is of the order of 5 nanometer, how many tubulin units are added to the growing microtubule per minute? Okay, so this is from your cytoskeleton part. Yes, right, Mahima, you are right. Lakshmi, clear, good. So try solving this particular question. Okay, here you need to solve. I'll give you all 1 minute time so that everybody can solve. Okay. Many of you all are giving me the answer as option 4. What about others? Okay. Still students are solving. I'll wait for some time. Okay. So now let's see what should be the right answer over here. So yes, the answer will be your 5200. Right? So the answer for this particular question is 5200. Now how will you solve this particular question? Now if first you should know what is a microtubule, right? I'm sure you all know. Now when you know what is the structure of microtubule, so you should know the microtubule is having 13 protofilaments, right? So like this, these are known as your protofilaments. So each protofilament is made up of tubulins. So it has two tubulins, alpha and beta, uh, the tubulins. So these tubulins are present as dimer, right? So now the question says size of one tubulin unit is 5 nanometer. So again, you have to convert it over here into meter. And the rate of formation of single protofilament is also given in the question, that is 2 micrometer per minute. Or you can convert it into meters or nanometers. So if it is nanometer, you can convert it into 2000 nanometer per minute. So number of tubulin units added per minute to elongate a single protofilament. So first we are talking about just a single protofilament. Okay, the rate is given and the size is given. So when you divide the rate by the size, okay, so that time you will get the number of tubulin units that have to be added per minute, right? So you get 2000 by 5, you get 400. Now, this is just for one protofilament. So, like that, you have to calculate for 13 protofilaments. So, you have to multiply this 400 into 13. Clear? So, you get the total number of tubulin units that is added per minute. So, because just 400 is just for one protofilament. So, you multiply with 13, you get 5200. Clear for everyone? Should I move ahead? Can I see a BIO if everything is clear? Okay, Vishal. Yes. Now, next question is the eighth question. So, this was asked in December 2017 to assess the impact of a newly identified drug when added to a culture of subconfluent HeLa cells. A researcher analyzes the fluorescence activated cell sorting FACS, that is the FACS profile of untreated drug versus treatment, that is no where untreated where drug is not there, versus treatment means drug is present. Okay, and they have given you two figures over here. So based on the FACS profile shown below, so you have to find out this drug inhibits which phase of the cell cycle. G1 phase, S phase of the cell cycle, third is G2, M phase of the cell cycle, fourth is G2 phase of the cell cycle. Okay. Some said fourth, some said 3. Okay. G2M. Okay, Vishal. What about others? Ritika says option 1. Okay, some said G1. Okay, then this is your S, this is your G2M. So you need to tell which one this drug inhibits which phase. By seeing this is when the drug is not present, and when you add the drug, this is the graph that you get. Okay. Okay. So many of y'all said 3 as the option. Many of y'all, some said 2. Okay. Amla said 2. Okay, let's find out. Okay, what should be the right answer over here? Now, whenever we talk about FACS profile and we are talking about the cell cycle, so how we get the graph? We get the graph something like this. You might have seen the graph. Okay, so this is your G1, this is your S, this is your G2M. So in this particular question, this is your G1. So here, they are not that much concerned about S. So here is the S phase that will be in between this G1. And then you can see the other peak, that is G2M. So this is how a cell cycle graph looks in your FACS profile. Now, in the first figure, there is the drug is not added. So this is 2n and this is 4n, the amount of DNA that is on the x-axis. But when the drug is added, so what we see is G1 phase is not there, right? But all the cells, so number of cells, you can see the peak is increasing, means all the cells, they are present in which phase? They are present in the G2M phase. So you have to find the drug is inhibiting which phase, right? This drug inhibits which phase. Now what we can say over here is, because all the cells are present in the G2M phase, means these cells are not able to exit mitosis or the M phase, because after the M phase only, after the G2, the next phase will start after this, right? So they are not able to exit this M phase, means the drug is inhibiting the cells at G2M phase. That is why all the cells are present in this particular phase, that is your G2M phase. Clear? Okay, so what we can say over here, the drug is actually inhibiting the G2M checkpoint, right? So the drug inhibits G2M phase as all cells are in 2n stage, and you don't see any cells in your, all cells are in 4n and no cells are present in your 2n stage. Clear? Yashira, it is not option 1, right? It is your option 3 will be the right answer. Yes, Debanjan. Okay. So I'm sure you have understood how to solve this particular type of question that is related to FACS profile. Okay, so you need to see where the cells are accumulating, and based on that, and you have to also consider, okay, the size of the peak. Okay, so in this particular question, because the peak size is increasing over here, means all the cells are getting accumulated over here. They are not able to exit this particular phase. Because they are not able to exit, means they are getting inhibited over here. G2M phase of the cell cycle is getting inhibited. Okay, students. Yes, yes. Clear. So let's move ahead to the next question. That is question 9. Is again about FRAP. Okay. So fluorescence recovery after photobleaching is a method to estimate the diffusion of molecules in a membrane. Fluorescently labeled molecules such as, first, a receptor tagged with green fluorescent protein, GFP. Second, a receptor labeled with GFP which interacts with cytoskeleton. Third, a labeled lipid. Fourth, a labeled protein that binds to the membrane surface, a photobleached, and the recovery profiles. So these are the recovery profiles that you see in the graph. A to D were obtained to estimate their diffusion coefficients. So which one of the combinations is correct? So you have to find. Okay, one is either, you have to say what is this. D is 1, 2, 3, 4, or C is what, you have to match. Clear? Got it? And you know what is FRAP. We already discussed now. You know what is diffusion coefficient. So try and tell me what should be the right answer over here. Okay, I see one answer coming up. What about others? Try and tell me what should be the right answer. Okay, I got another option. Some said 2, some said 3. Think properly, try and tell me. Okay, Sunanda, Akash, Soni, Gunu, many of y'all have answered. Still, some students are answering. I'll wait for some time because it is, you have to think properly, you have to match them properly. But just apply the diffusion coefficients that we understood, okay, and the different molecules, fluorescent labeled molecules. So it's not very difficult. Okay, so let us see what is the right answer over here. For this particular question, the right answer is your option 2. Okay, now, so what it means over here is recovery profile, okay, will be least, means the time for recovery will be least for that particular molecule, the fluorescent labeled molecule, which has the highest diffusion coefficient, right? Right? Recovery profile will be, the time for recovery profile will be least for that particular fluorescently labeled molecules whose diffusion coefficient is the maximum, right? Means more diffusion coefficient, more fastest it can move, so recovery time will be less. So in that case, here, if it is simple a labeled lipid, right, so in that case it should have the maximum diffusion coefficient because it is free, right? It can easily move. Clear? So that is why B, you can match it with 3. Okay, now fourth one, what, so A, the next recovery profile is for A that they have mentioned, a labeled protein that binds to the membrane surface. So when you compare this with the labeled lipid which is free and the one that is bound to the membrane surface, so in that case definitely it will have a little lesser diffusion coefficient. Okay, that is why it is taking little more time, recovery profile is taking more time. So A, you match it with 4. Now what about the other two options? Now when you compare, okay Aditi, you have no idea, so let's try to understand. Okay, so when you compare the first one, a receptor tagged with green fluorescent protein. Now here the question is saying it is a receptor. So already it is a receptor. Okay, so when you compare it with a receptor and a lipid and a protein, so receptors will have many transmembrane domains because it is a receptor, it is present on the surface, it will have the transmembrane domains, it will, it will span the membrane. So again, what we understand over here, if there are more transmembrane domains, means it will show more recovery time, right? So more recovery time means maybe the one can match with D. Okay, so but here what is our answer? Answer is option 2. So already we have got, because we are in the question, the options that they have given is for the fastest recovery. So B will show the fastest, that is lipid, and then the other one is the one that is just bound to the membrane surface. But if it is a receptor, it is spanning the membrane, so it will take a lot of time. Okay, and then the second option says a receptor labeled with GFP which interacts with cytoskeleton. So you can imagine if this is your no, the membrane, so it is your double membrane, so you have the lipid and then there is a receptor over here and it is interacting with the cytoskeleton. Okay, maybe a microtubule or microfilament in the cytoplasm. So you can imagine, so this will take definitely more time. Okay, or the recovery will be, the least. Clear? So 1 and 2, we are not concerned about it because if you see, the options are based on the fastest recovery profile. So B will match it with 3 and A will be equal to your 4. Clear, students? Aditi, I am sure now you have understood how to solve this particular question. Okay, just that everything is given in the question, you have to match it and you have to try to find out what should be the right answer over here. So the right answer is your option 2. So if it's clear, I will move ahead. So we have one more question. So let's see. The 10th question says, during the cell cycle regulation in eukaryotes, there are post-translational modification. Okay. No, it will be option 2. Okay, there is a query over here. Okay, B, it cannot be 1. So this is option 4 is wrong. Okay, B has, because 1 is a receptor. So it will not show high diffusion coefficient. So its recovery time will be more. This recovery time will take more, right? Okay, so that is why fourth is not the right answer over here. Got it, Vishal? Are you clear with it? Or any other queries is there? Okay. Point 2 matches D. Okay, yes, in that way you are right. In that way it can match with D, yes. So that is what I said. We are not concerned about these two because the question, you can see the options. So we are more concerned about 3 and 4, but yes, you can match it like this will be the least. So we can expect that it is D and this can be C. Yes. Got it? Okay. So good to see that you all are able to understand now. Let's move ahead. I'm sure if you get a question like that, you will definitely be able to solve it. Okay. Let's see the last question. During the cell cycle regulation in eukaryotes, there are post-translational modification of protein factors, which act as switches for different phases of cell cycle. A cell population of yeast was transfected with gene for V1 kinase, modify CDC2 protein. Assuming that the transfection efficiency was 50% only, which of the following graphical representation of the results is most appropriate? So you have 4 graphs over here and you need to tell me which one is the most appropriate. Try and tell me what should be the right answer over here. Okay. So you need to consider what, no, you should know what a V1 kinase does. Okay. Only then you will be able to solve this question. Y-axis is cell number. Okay, y-axis is cell number, x-axis is time, and there are two, the one line where there is the dark dots that you can see, the filled dots, those are transfected ones, and the one that is not filled up, those are non-transfected ones. So you have in the graph you can see 2 lines are there. Okay, so I see some answers coming up. Okay, so we'll see how to solve this particular question. So first, if it is a V1 kinase, okay, so what it does, tell me what a V1 kinase does. It is a kinase, so definitely it will add a phosphate, right? So if it is adding a phosphate over here, so if it is a V1 kinase, it is adding an inhibitory phosphate, right? So when it is adding an inhibitory phosphate means we can say that wherever the V1 kinase is getting transfected means there will be, so you can see if V1 kinase is present, so it is still inactive. So mitosis will not occur. Yes, Lakshmi. Inhibits cell division. Okay. Until in, yes. So over here, what is happening in this question is why it is option 1, because so the answer we saw, it is option 1. Now why it is option 1? Because the question says a cell population of yeast was transfected with V1 kinase, assuming that the transfection efficiency was 50% only. So transfection efficiency was 50% only. So now the ones that are non-transfected, so there the cell number will increase. But where the cells are transfected, so there we will expect that the cell number should decrease. But the question says it is just 50% efficient, means there will be still some transfected cells, okay, where the transfection is not efficient enough. So they will be able to divide, right? And that is why you see that the cell number is increasing, but it will be lesser compared to non-transfected cells. Yes, Antara, right, not 100%. Yes, so it is 50%. That is why the right answer is your option 1. Okay, So you can see now, you can tell me, what do you think about the unit 2 questions? Easy, difficult, need to practice? Yes, only right. If it is 100%, so we'll go for the next graph. Okay, that is, you can go for option 3. Okay, graph C. Moderate. Okay, yes. So what we see over here is, if you practice properly, if you know how to solve the questions, if you have the idea about the various topics, then with practice you can master this particular unit. Thank you so much. I hope you all have enjoyed the session. Okay, so stay tuned, be enlightened always, and you can join the next session that will be on Monday for unit 3 and 4. Thank you so much. Yes, you need to revise, Sunanda. Yes, needs practice. Yes, I agree with you all. So keep practicing. Yes, Antara, the mathematical questions you need to practice. Okay, keep practicing, and I wish you all the best for the upcoming exam. And the ones who are not able to solve, don't be disheartened. Okay, you can join the upcoming batches, and then you can practice and you can be successful. Thank you so much.